Okay, so let's start off the class today. So hope you all have the nice week, and also the Monday and Tuesday. This is quite, the lovely the semester starts. I mean, because of weather. I mean, it's a bit disappointing because we cannot meet at the campus. But yeah, but anyway. Hopefully you have really enjoyable time in your place. So before we go into today's class, I have one short notice about the second week of the whatever the our department introductory course, because I mean, as you know that I'm the lecturer of the week. So the notice for this is not relevant to this genetics course, but this is the Friday class of the other class, but I mean basically the same lecturer me that time. So I put the pre-class the questionnaire, and I mean if you get the link from the Blackboard, because I put the announcement tab, and you can find the link for this. And if you submit your questions about, my class or the class in the next semester, the biostatistics, or anything about like, bioinformatics or genomics Korea, if you think about that, please leave your question. Then I can follow that questions during the class time. And this is not the English class, so I mean I can follow in our language. So I mean that is good, right? So so please submit this question to using the link on the Blackboard. Okay, so and then there are some questions on our Slack. Let's go to Slack. So so two questions from Minsa. So one question I can handle before the class. So the question was, what is the differences between the hereditary and inheritance in the context of genetics? So the answer is they are basically same meaning. So I mean there's no huge differences between these two, and also the words is interchangeably used in the same context. So I mean I would say these are same, but technically on the dictionary, inheritance is more broader, the meaning, containing the meaning of hereditary. But I mean, as to the genetics context, they are basically same, but somewhat what I found is the inheritance is more formal and also more widely used in the latest genetics research rather than heredity. So, I mean, but I would say it's technically same. But, when I prepare the slides, I try not to use the same words because it's too broad. So sometimes I use this word in this context, this word in this context. So, but I mean, this is a good question because you can keep in mind that some of the the context I use the sort of use different words for the just not to use the same word. Okay. So this is good. And the second questions, the set of questions from Minso, I can follow up after the course. Okay, so back to our slides. So today we're going to talk about the biometrical genetics. So the biometrical, probably quite awkward words because you have never experienced these words during your biology learning. The biometrical, literally the area of measurement of the biological activities. That's more like a dictionary, the definition. But biometrical genetics is that the people who use the sort of mathematical and statistical notion to summarize the genetic activities. So we are going to learn about that sort of the camp of the math and stat people in genetics history and how they evolved in late 90s and how they conflict with the questions arising from the Mendelian people. Okay, so what we're going to learn today, we got three things. So first one is the conflicting views between the Mendelian and biometrician. And the second one is the how biometrician emerged at the time. And the third one is the Fisher's genetics model, which is, I would say, the classic model we have to understand in modern genetics. Okay, so so the first topic, so conflicting views between the Mendelian and biometrician. So as I brought last time, we had a question about the height, right? So if you think about the height, how can we explain that traits using the Mendelian perspective, right? So we know that the height is somewhat heritable because the tall parents get the tall child, right? So we would say height is probably heritable, and also the genetics is working in that traits. But we know that the height is quite continuous and quantitative trait, which kind of explained by Mendelian factors, right? So Mendelian factors is the binary characteristic, and we can always, you know, they compare quite distant characteristic between between individuals, right? But height is something different from that. So the question people had at the time was, do quantitative traits have the same hereditary and also evolutionary properties as discrete characteristics? So that was the question at, you know, arising in late 90s and early 20s. So the famous historical event, we called it the height and P debates, which happened at the early 90s, the 20th century. And that was the debate between the Mendelian people and biometrician people. So as I mentioned, that the Mendelian is the people who claims Mendelian genetics is working in inheritance, and Mendelian genetics rule out all of these genetic phenomena. And the biometrician is the people who claim traits are heritable and also continuous. So this is somewhat similar to blending inheritance proposed by Charles Darwin. Okay, so let's quickly recap what Mendel proposed at the time in his particular inheritance model. So Mendel did experiment with the peas, and he used the hybridization experiment, and he proved that the heritable factors is discrete particle factors passed on the intact from parents to offspring, right? That was Mendel's view on the genetics, right? And Mendelian genetics is we can say it's a reductionist way to formulate inheritance pattern. So if you think about the Mendelian rules like segregation, assortment, and dominance, that was the clear sort of formula to explain this inheritance pattern, right? And this Mendelian rules is rediscovered in 19th century by the findings in cytology, right? So, I mean, we can clearly get the examples of the P color then explained by the binary trait by Mendelian factors, right? Then Darwinian blending inheritance is somewhat different from Mendelian perspective, right? So what is Darwin? Darwin's idea? He tried to explain the evolutionary properties and variability of traits within a population. And he tried to explain that the variability of traits is heritable, and that is the major sort of driving force and phenomena happening in the evolutionary process, right? So he tried to explain the situation using the natural selection perspective, and then he proposed the idea of how that variability inherit to the next generation using the model of the blending inheritance, right? And as we know that this model was a provisional theory for his evolutionary insist, and then and according to this blending inheritance, the offspring traits or phenotype is an average of both parents' traits, right? Okay. So what biometricians claim on the traits from this the Darwin's idea, because the biometrician is mainly working in the British Society late 90s, and they are, I mean, naturally interacted with the Darwinian perspectives, right? And also Charles Darwin as well. So biometrician is inherent their idea about the blending inheritance model, and also think about the sort of gradualism and the trait variability, right? So biometricians think that the traits are continuous and the traits are not binary, right? So this is basically the same notion as the blending inheritance. So so we can say that the Charles Darwin's idea is definitely influenced on the emergence of biometricians like Francis Galton or Carl Pearson. And biometrician, as I mentioned, that is a biometrical genetics concerned with the inheritance of quantitative traits. And their view is the traits are continuous and heritable. So if you look at the height, height has to have the values following some distribution in our population. So there's no, way to, the height is just recessive or dominance as Mendel proposed, right? So height is a trait, has some distribution. So now we know that this is following the normal distribution. But anyway, so the individuals in our population has a continuous trait as height, and that is sort of the natural phenomena we can think about the genetics in our population. And biometricians' view was that most traits are actually continuous, and also these continuous traits is the key things we have to implement it in our genetics model. So, I mean, their view is naturally arguing the importance of the blending inheritance in this the genetics and inheritance model. Okay, so let's talk about the sort of the details of how biometrical genetics has been emerged at the time. So first person we can think about is the Francis Galton. So Francis Galton is a Victorian mathematician, but also people say polymath. I mean, polymath is that the person interested in so many things and has an intelligence in so many the field and influence on so many things as well. But anyway, so Galton is the person who pioneered that the quantitative traits or quantitative nature of the genetics in our population. So he basically looking at the quantification of resemblance between relatives for traits like height using regression and correlation. So more background on the Francis Galton. He's a cousin of Charles Darwin, and he actually contributes so many different things on early genetics and biology, because he's sort of totally obsessed with the quantitative nature of the biological mechanisms, and also try to put that phenomena into the mathematical and statistical notion. And also Francis Galton, as I mentioned, he's a polymath, so he is influenced in many different science. So, I mean, if you're interested in that, please go to the link written by New Yorker, and the title is the Whenever You Can Count. This is just brilliant. The article we kind of see in a Korean journal. So you can know much about the Francis Galton's the life at the time. So the Francis Galton is, influenced many things. I mean, I would add extra information on that. He developed some idea about the weather forecast. So some of the mathematic formula we used in the weather forecast prediction is actually based on the Francis Galton's idea. And also the fingerprints. We use the fingerprints for our smartphone or somewhere in the door access things. Using the fingerprint idea is actually developed by Francis Galton as well. So he's definitely many talents on that. And also some of the mathematical format, so regression or correlation, is actually initially developed by Francis Galton. And his students, his follower, the Carl Pearson, developed this idea much more elegant way and proposed in a mathematical format. But the prototype is actually from the Francis Galton. So Francis Galton is actually influenced many things. But why we didn't know anything about Francis Galton? Because he's a source of eugenics in early and mid 20th century. So if you look at his book, like A Natural Inheritance, written in the late 90s, you can search this book on Google and you can read the original text, and that is really politically incorrect. and I mean, there's no way we can, sort of respect or we can see, as a person in a scientific, virtue. So Francis Galton has really bad reputation on that. So I mean, we ignore his, scientific achievement, but actually so many, the scientific things actually start from the Francis Galton. But anyway, okay, so Francis Galton is interested in sort of the quantitative properties in underlying the inheritance, right? So he got into sort of the study of the inheritance model, and he looked at sort of the trait variation in population and which form of that variation resemble which sort of the mathematical perspective. And he collected the data and he tried to explain by the mathematical idea. Okay, so what Galton see on trait variation is that the traits like height, weight, or even like arm length, they are normally distributed, not binary, right? So this is sort of the contradictory point as Mendel's idea, right? And also what he thought that the traits are normally distributed, right? This normal distribution indicating that the traits has to have mean value within a population, right? So we can summarize the population values using the mean, right? And also the concept of the variance, right? That is the deviation. So we can think about the how much the individual has deviated in terms of the trait value, right? And also the traits can be transformed to the mathematical situation, right? So normally distribution means that we know the distribution in generally working in many different situations. That means that if the traits follow the normal distribution, then we can just put it into that, then using that the mathematical transformation into our calculation, right? And Francis Galton, as I mentioned, he introduced so many essential concepts of the mathematical situation using this genetic idea, including like regression, correlation, and that is all, the good framework to study continuous variation in many different organisms. Okay. Okay, so what Galton measured at the time? So let's look at the height measurement at the time between the parents and offspring. So as I mentioned, that Galton tried to measure everything as possible. He knew that, right? Okay. Oh, okay. So I got some message from other students. Yeah, that's fine. Okay. So if you cannot access to the internet right away, you can just reconnect it, because you can allow to come into this session without my permission, right? So I mean, I'm open to that. So I mean, you don't have to excuse to that. That's fine. Okay, so back to the Galton's measurements. So Galton measured the height of the parent and offspring. That is quite, it looks simple at this time, right? But think about the situation in 19th century. Having a thought on, okay, let's measure the height of parents and offspring. is something odd at the time, because I mean, people are not really interested in those activities, and also put it into the numbers, transform into the numbers is something bizarre, right? I mean, people not really want to do that. But Garten, he tried to, put it in many different phenomena into the number situation, and that was the sort of the great start of the genetics model, even for nowadays, right? And what he found that the mid-parental height has almost no deviation to their offspring height. So in other words, mid-parental height is the nearly identical to offspring height. So if you look at here, this is y-axis is the mid-parent height value. So we get the average height value between mother and father, and we get the value. And if you measure the height value in an adult child, not the young child, but right, because the height is grown up. So you use the adult children. So if you get the tall mid-parent value, then you will have the tall child, right? And that is almost identical between his measurement, right? So he tried to explain the concept using the lines he drew from this data set, and he tried to measure the deviation between his predictions, so between the parents and children. And he explained that there is almost no deviation between parents and children pair. That means that the height, we can say that this is the heritable between generation, right? So as I mentioned, that the traits are resembled between parents and offspring. So as I mentioned, the tall parents likely have tall offspring, and that is the ultimately introduce the concept of the correlation, because these two things are quite similar to each other. Okay, so and then Garten also using other types of experiments. So he also used the sweet peas as Mendel, and he did the experiments. So what he did is he looked at the sweet pea size between the mother and daughter seeds, and look at whether there is any correlation between the parents and child, as he observed in human height, right? And what he did is he distributed the packets of sweet pea seed to 7 different friends, right? So there is like a random distribution. And then he measured that the size of the seeds after the one generation. Then he calculated that whether there is any seed size similarity between the parents and the daughter generation. And that was as you can see from here, the parent size and daughter size is also quite correlate to each other. This is the same observation as you see in the human height data set as well. Okay, so I mean, from this experiment, Garten was quite confident about other than human height or human trait, even the traits in a natural population like peas is also heritable, right? So he was quite confident about the traits are continuous, and also these continuous traits are heritable, and these continuous traits are preserved across the generation. That is the same idea as Darwin proposed in his natural selection model, and also blending inheritance, right? Because the Darwin also observed that the traits are variable across the individuals in a population. And then Garten clearly see that those variance, those variability is preserved across generations, right? Okay, but in Garten's idea, he used the more mathematical notion and also more formula, and he clearly explained that how much the similarity preserved across the generation. Okay, so Garten's experiment and Garten's arguments actually influenced so many different, mathematicians and statisticians, including Carl Pearson, Walter Walden, and those people are the founders of modern stats and modern mathematics. And these people is totally shocked by the Garten's genetics idea and Garton's measurement and use his data set, use his experiment, and they are trying to formalizing the Garton's idea into the mathematical model. So what Carl Pearson did, he invented least square models of determining the regression line. So he used the concept of the least squares to measure the deviation between the parent and child trait value, right? And that is the sort of the start of the regression idea in statistical field. And also Walt Walden has found the mathematical and statistical journal called Biometrical. This is one of the famous journal in nowadays as well, and that is the aiming the promoting the statistical studies of biological phenomena. So I mean, I would say Mendel's idea is also quite mathematical and also quite elegant experiment, but also in the meantime, there is a notion to develop sort of or elaborate the mathematical and statistical framework in a biological study. That is quite, the important things I'd say, because, if you study chemistry or if you study physics, the mathematical or statistical concept of the physics or the chemistry is way ahead than biological science. The reason is that the physics or chemistry is actually born to be like that. And they try to set up the principles and they try to set up the formula and they describing the phenomena using the principles or formula, right? And then they're more precisely working in their observation. So more technical or the mathematical advanced is way ahead in those fields. Compared to physics or chemistry, biology is way behind that notion, because the biology is based on the observation, and that observation has to be very subjective, and observation has to be very limited, because we only describing some samples within the entire population, right? And also the biological phenomena has many exceptions. So that's quite different perspective compared to chemistry and physics. But the notion is quite changed dramatically in last decade, because now the development in a technology and also the data science computation and also the biological techniques. And nowadays we are taking that sort of the mathematical and statistical movement in biology. And that actually driving so many things in the biological science. So think about the COVID-19 vaccine, right? So if we are in the 1920s, like when, the Spanish influenza outbreak, we couldn't do anything. So we means that the biologists do anything, right? We can only do like, we can only do, wear a mask, or we can only do, avoid from the affected people. That is the only thing we can do that. So that time clinicians or doctors has much more, the authority in terms of the prevention and also the public movement. But COVID-19 is different, right? So COVID-19 is outbreak a year ago, and during 12 months, we developed the vaccine, we developed the way, we sequenced the virus RNA, and we tried to come up with how to design vaccine over the 12 months. That is great achievement, right? But to think about the situation, how we can discover these things right away, because we developed a new technology called genomic sequencing, right? That's why we sequenced COVID-19 RNA sequence within a day, right? A day. And then we get the sequence and we assemble the sequence into the developmental format. and that only takes a day as well. And then people try to develop the proteins we can use in the vaccine, or to develop the RNA template we can use in the vaccine. That only takes like 4 months, right? This is amazing advancement over the time, right? And now we know that the many of the disciplines in biology is actually driven by the situation where we take advantage of the mathematics and statistical formula into describing the new phenomena and also developing the new product, right? So please keep in mind that you can learn from the history, right? So when we think about how we describe the genetics, how we describe the heritable phenomena in a human population, you can think about the Hippocrates or Aristotle. They only think about within their minds, right? Only just an arguing, right? And Lamarckian is same. But Mendel did the experiment, right? And that was the great achievement because he hands-on something and generate the data, right? And the Galton, Carl Pearson, all these people come up with the mathematical formula and they put it into very reduced dimension to explain this situation. And that is sort of the advancement over the decades in late 90s and early 20s. Then back later, we coming up with the better technology and see many different things that will be followed in later section. But anyway, okay, so back to the Carl Pearson idea. So Pearson and Lee, the paper 1903, they tried to formulate all the trait correlation within a family, right? So what Carl Pearson did, he measured various physical traits like height, arm length, and he tried to put it into correlation calculation. Then he proved that most of the traits are correlated within a family, and that clearly indicating that the traits are heritable and also the continuous traits, right? Okay, so before we moving on to the next session, the next slide, if you have question, you can leave in a chat, but don't send me the direct message. You can leave the question on the public, right? So I got the question from Liu and Lee. Don't send me to the direct message. I really argue with you that the questions has to be on the public, right? Questions has to be shared by students, right? Otherwise, there's no way we can do the education, right? So please leave your question onto the public place, right? So that is the part of the learning, right? So don't send me the direct message. Okay, so back to the Pearson Lee. So this is the diagram what Pearson and Lee did at the time, right? So he used he developed the experimental the measurement in his experiment, and he measured the arm length and also the arm width according to many different physical places, right? Okay, then he put it into correlation calculation. Then he drew the hand lines and see whether these traits are correlated or not, right? So as you can see from here, Pearson's the traits are correlated between the parents and children, and there is quite high similarity. And they got to know that the 50% similarity between the parents and child. And now we know what 50% means in the similarity, right? Because the father and mother share half of their genome with us, and that's why we have the 50% correlation with our parents, right? We know that that's the background, right? Because according to Mendel's segregation, we get only one part of the alleles from one of the parents, right? And we get the rest half from the others, right? So this 50% similarity, we can clearly explain that situation. But at the time, Pearson and Lee didn't know what 50% exactly means in this biological situation. So because they I mean, unfortunately, they didn't know anything about the Mendelian genetics and Mendelian interpretation. Okay. And then the Carl Pearson's idea is exactly following the notion from Francis Galton's experiments. But people also at the time start to see the concept of Mendelian genetics in British society, and people start think about how Mendelian genetics is complement with this biometrician idea. So some of the biometrician definitely totally opposite to what Mendelian proposed. So Mendel genetics is total bullshit, and we don't, take that concept. But some people, I mean, other people, they think about, okay, so probably Mendelian genetics can be working with the biometrician's idea. And the first person think about that unified model was Brownlee. So Brownlee's 1911 paper, he provides sort of mathematical models how Mendelian genetics is working in the biometrical genetics, like the situation of continuous traits like height, weight, or blah blah blah, and how Mendelian genetics is working. So what Brownlee proposed at the time was the combination of multiple Mendelian loci is actually resulting the continuous traits. And this Brownlee's idea is the forerunner of the Fisher. So this is like a prototype of the Fisher's model. Okay, so what Brownlee's insist at the time. So let's think about the 50% correlation we observed in the Pearson's paper. We know that the 50% of correlation of traits because you transmit 50% of your genome to your child, right? This is more of the words we think in these days. But anyway, so 50%, we know that this is the sort of the background similarity we can transmit between parents and offspring according to Mendel's segregation idea, right? And you get the 50% similarity between this generation. Then you get the also 50% similarity between these two generations. Then you can say that your similarity between the grandfather and child, or the you and grandchild, is the 25%, because you get the 50% transmission and 50% transmission. That has to be 25 probability of the transmission. Then we can say that the backgrounds of the similarity or the correlation is the 25%. So Brownlee took this as a blending inheritance. So in his model, so we use the examples of the number of generation determining the correlation coefficients, which is the whatever 50% or 25%. But if you think about that generation, and if you replace the generation with the number of loci, right? So loci is the plural form of the locus. So let's say you have one locus, you will have the 50% similarity or correlation coefficient. But if you have two loci, then it's going to be 25% because it has to be 50% times 50%, right? So your correlation coefficient is going to be 0.25, which is the 25%. So if you have more and more loci, so if you combine them more and more, then your correlation coefficient has to be diluted as the number of the loci you used in that the calculation. So Brownlee thinks that this is the same situation what Darwin proposed in his blending inheritance. And also this is totally complement with the idea of the Mendelian genetics. And this is followed by Fisher's idea in 1918, but Fisher's idea add on the clever things on his model. Okay, let's look at that. Okay, so we're going to discuss about the last, the sort of the major topics in this class is the polygenic model. And we start off the Fisher's model, 1980s. So Fisher proposed his idea on this paper, 1918, and this is the core paper in genetics history. And let's summarize what he proposed at the time. So what Fisher showed at the time was a quantitative trait could be explained by Mendelian inheritance if several genes affect the trait, right? This is the key sentence. So quantitative trait means that the continuous trait we see in height, right? Then those traits can be explained by Mendelian inheritance, which is the binary factors, right? And why he explained that this situation, we called it the polygenic inheritance. The reason is the poly means the many, and genic means the genetic factors. So let's say several genes affect traits. That means that the several poly genes affect traits, right? So that situation we call the polygenic inheritance. And that is the sort of underlying mechanism for quantitative traits, right? Then Fisher come up with the idea about the additive and dominant factors, right? Then also coming up with the idea about the resemblance between relatives due to their genetic covariance, right? Okay, and this Fisher's 1980s paper is unified the idea between the biometrician and Mendelians. And this is the key things we can understand the modern genomics in these days as well. Okay, so let's look at the details of Fisher's model. So we have to start with the Fisher's single locus model. So this is one of the key things. So let's assume that we don't know anything about the dominance between two alleles, right? So in other words, we don't know anything about the dominant alleles, right? And also we assume that locus follow either dominant or additive, right? So locus means that the position where allele exists, right? So we can just specify limit one locus at this time. Okay, and we have to determine whether the locus follows dominant or additive, right? So what we can do, what we can have is A1 allele or A2 allele. So it can form A1 genotype, A2 genotype, or A1 A2 genotype. Okay, so let's put in this way, and let's think about if you have heterozygous, which is A1 and A2 genotype, and it has the phenotype value somewhere in between 0 and A, right? So let's say you have the phenotype to minus A to A, and this phenotype is somewhere between the 0 and A. Then in that case, we can say that A1 has more effect than A2, right? So we can say that if the D or delta is greater than 0, then the A1 is the dominant allele to A2, right? But if you have the opposite situation, which the D is smaller than 0, which is the delta is smaller than 0, then A2 is dominant to A1. So this case, A2 is dominant allele, A1 is the recessive allele, right? But let's say if you have delta is exactly A or exactly minus A, that case you can say you have complete dominance, right? So you can say that A2 is totally affecting on this situation. So this case, A2 is complete dominance, or A1 is the complete dominance. And this is the situation we can see in the Mendelian dominance heterozygous genotype consequence, right? So Mendelian genetics, we explain this as a the heterozygous has the dominant value in F2. That case we explain, F1. That case we explain the complete dominance. But in real world biology, it's always not happen like in this way, not like in the Mendelian way. Okay, so you have to know about the differences between the three, right? And then once you have the value is greater than, there has to be great. Yeah, I'll fix that. Has to be greater than A or minus A. There has to be over dominance, right? So this case, A2 allele has the overdominance effect, or A1 has the overdominance effect as well. Okay, but the situation is if you have A1 and A2, this heterozygous genotype has the 0 phenotype delta value. That case, A1 and A2 is additive, right? So additive means that A1 and A2 has the same equal consequence in phenotype development, right? So that case, we called it additive, right? So the situation in polygenic model, we are assumed that the locus follow the Mendelian rule. So that means that the binary factor A1 and A2 allele we have, and we assume that there is no way we can think about the dominance between these two alleles, right? Okay, and according to that model, and in a single locus, we can have the 3 possible genotypes: A1, 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 A2, A2, and A2. And the ratio between the 3 genotypes is 1:1, right? This is the obvious one. And let's assume that you have the 2 loci at this time. So you have A1, A2, one set of the alleles, and the second set of the allele is B1, B2. And that is the independent loci, right? Independent locus. So one is the first locus and the second locus. In that case, I mean, again, we don't know the dominance. We don't know the we don't know the dominance, and also that one is also following the Mendelian rules. In that case, we have the ratio 1:4:6:4:1, right? This is again quite obvious. And let's say we have the 3 loci this time. That is A, B, and C, right? So 3 different positions. And we again have the Mendelian factors, and also we don't know about the dominance between alleles. In that case, you have the ratio is 1, 6, 15, 20, 15, 6, and 1. And that is these types of the genotype values. And you can see from this 1 locus, 2 loci, and 3 loci situation, you will see that the distribution of the genotypes is getting some distribution. It's like a normal-like distribution, right? So Fisher's idea, as Brownlee's idea, when you add a large number of loci, then you get the normal-like distribution. So each distribution, each canton here, you can say that this is the equal to individuals with certain genotype, right? Because the individuals like me and you only have one genotype, one of these, right? So if you combine multiple loci, you will end up sitting in somewhere in here, right? So let's say, if you think about height, and let's say A2, B2, and C2, if you have this combination, so you will be here, then you're gonna have like a 200 centimeter height, right? But you have A1, B1, and C1, then you have 140 centimeter height, right? Because how you combine your alleles across many different loci, then you will have certain genotype, then that genotype going to determine your phenotype values, right? And if you think about it, if you add up more and more loci in that situation, then that trait's going to have some normal-like distribution, right? But think about the situation. So let's say in the human genome, we have millions of loci in our genome. I mean, actually billions of loci in our genome. Then you add up the billions of allele sets into the trait calculation. Then you have quite almost the quite continuous normal distribution in your height distribution, in your height, right? But if you think about the height doesn't have the interval, right? This is the key thing. So let's collect all individuals in the world, in the planet. then you measure the heights. The heights has, some intervals because we have definite numbers in the planet, but it actually has almost infinite distribution in the traits, right? So what I want to say is that how we explain these finite intervals using the finite numbers of the loci in our genome, right? Because our trait is almost infinitely continuous in their distribution, right? So what Fisher came up with the brilliant thing is that he add up extra factors, additional factors, that is environment, right? Environmental factors, because during your lifetime, environmental factors is almostly infinite numbers, right? Because we can guess the environmental factors, how much you sleep, or how much you do the stretching, how much you do the exercise, right? But we can actually add up so many things, so many, the physical activities or, environmental exposures or something, right? So during our lifetime, during our development, environmental factors is almost infinite numbers. So if you add up the environmental factors like a binary, like whether you're exposed or not, then your height distribution, it gets infinite intervals, right? There's no intervals, almost no intervals in the traits. So if you think about like a weight, or if you think about, mental activities, the level of depression or level of happiness, all of these things doesn't have intervals in their trait distribution. So when you think about the traits are influenced by the number of genetic loci, but also influenced by the number of environmental factors. That is the key concept Fisher proposed in early 20s. So Fisher's model includes multiple compartments explaining phenotype variance. So multiple compartments means that many different genetic factors and also non-genetic factors. So what is the genetic factors? These including additive factors, dominance factors, epistatic factors, right? So I'll explain what is the additive, dominance, and epistaticity in this context. I'll probably come up with better examples in the later section. But anyway, you have to remember that you have to understand that there are many different genetic factors accounting for this continuous trait, and also continuity is established by the infinite number of non-genetic factors. Some people say that is the environmental factors, but I think it's technically incorrect, because we should say this is the non-genetic factors, because some of the factors is actually stochastically happening in our body. So stochastically means that they're randomly happening in our body. And that is also the driving factors, the phenotypic variance. So we have to keep in mind that the non-genetic factors is more precise term than environmental factors. But conventionally, people just say environmental factors. But anyway, so Fisher's model has the compartment things. So once you have the trait, the variability, like the phenotypic variance, then you have interactions between the genetic and environmental factors. And that is the classic formula, which is the phenotypic variance P equal to genetic factors plus environmental factors plus genetic factors interact with environmental factors. P equal G plus E plus G by E. This is the classic things we have to understand in modern genetics. Okay, so Fisher's partitioning variance model introduced so many key concepts in genetics, especially heritability. So heritability is the concept summarize or calculate how much the variation, how much the phenotypic variability is explained by genetic factors, right? So heritability is the framework we can measure the differences explained by genetics. And we're going to learn about how to calculate that one in a later section. Then Fisher also influenced to introduce the concept of genetic architecture. So genetic architecture is the key term I would pick up in 20th, these days genetics. And genetic architecture means that the composition of various genetic factors upon a phenotype. So if you look at the same phenotype, it actually has many different genetic factors in the trait, right? And nowadays, clinical genetics or medical genetics is whatever the genetics is interested in, how much the genetics is influenced on the trait or disease, and also which factors and what types of factors is influenced on the trait, right? Okay, so genetics is the science to identify genetic factors associated with trait and disease, but also the study of the contribution of the genetic factors. So we can talk about the sort of common misconception on genetic studies, and also the common misconception I get nearly every day, because some of my friends or my acquaintances ask me, what are you doing for your living? And I said, I'm a geneticist, and they say, wow, you're a geneticist, and whether do you know the baldness is genetics or not? Then that question is actually ridiculous because the genetics is not about identifying whether the trait or disease is determined by genetics or environment. It's actually the field of the study how much genetics or how much environment explain that trait, right? So the question like, is baldness caused by genetics? This is totally wrong question. That is the wrong. It's already wrong by early 20s Fisher's model. The more precise question is how much genetics can explain baldness, right? We have sort of several same examples like intelligence or height, right? So height is genetics, or intelligence is genetics, right? I mean, we are studying how much genetics influence intelligence or height, not deterministic model, right? So don't be fooled by sort of the dichotomy concept of the genetics, right? So like nature versus nurture. This is quite famous analogy by Matt Ridley, bestseller books in somewhere in 2001 or 2005, something. You probably read for your university exam. But I mean, this is Matt Ridley is not scientist. He's just one of the just a public whatever writer. So his conception about the nature and nurture analogy is actually borrowed from the Galton's book in Hereditary Genius in 1890s, because Galton used that terms from the the William Shakespeare's book The Tempest and Devil, a born devil on whose nature nurture can never stay, and he brought the sentence to his book, and it became quite famous sentence in genetics. But this sentence give us quite many common misconception in our field, and you have to remember that the genetics is not dichotomy things, and we have to consider sort of the continuous and polygenic perspectives in our study. So why I care about this? Because most traits and diseases are actually following the polygenic model. So that means that one gene or one allele unlikely contribute 100% to phenotype. That is for clear. And the one allele can contribute very little. But how much little, we don't know. I mean, it depends on the disease, it depends on the mutation, it depends on the alleles. I mean, alleles is equal to mutation, so I accidentally say mutation, but anyway. But the problem is that the modern biology is heavily based on the molecular genetics, and molecular genetics generation is heavily based on the monogenic concept, right? So, I mean, during your university or even if you go to graduate school or even like, the fancy company, and, people still think about the monogenic perspective in trade, even the medical schools too, and pharmacy, pharmacological science as well. but this is actually incorrect, right? As we see more data, as we see more and more population data, we know that this is wrong, right? We come up with that, the counterexamples, why this is wrong, and we come up with that things many in later section. But you have to understand that the most trait and disease we study in these days are actually following the polygenic model. And you have to think about your, the groundings out of this polygenic situation. The reason is that, as I mentioned, that nowadays, over a decade, we have new biological technologies like genomic sequencing, cloud computing, whatever the machine learning technology, the new types of the fancy algorithm. We are seeing totally new dimension of the biological traits and biological phenomena. And now we can interpret the quantitative nature of the biological activities. And these things clearly indicating that the polygenic model is correct, right? So we come up with more examples in later section, but keep in mind that and also think about the situation. Okay, so you can read about the summary. And also I put quite brilliant papers written by Peter Vischer. Peter Vischer is the modern Mendel, right? So he's the living Mendel in these days. He's the amazing scientist. He's an amazing geneticist. He's just brilliant guys. And I borrow so many terms from his papers and YouTube lectures. I actually, earned my PhD from his institute. But yeah, Peter Vischer is the amazing person. And I highly recommend reading these articles and expand your thought and expand your, the perspectives toward the polygenic models. Okay, so before we heading to the close, let's look at the questions from Minza at the in the Slack. Okay, so the questions is actually written in Korean, so I can quickly recap with English. So she asked that I don't know you are sure he, but anyway, this person asked that the locus is technically meaning the position of alleles on the chromosomes, right? Then I can say you are she, but she questioned that, her question was that whether this locus you can interpret in a Mendelian factors. Yes, yeah, you can do that. Yeah, at this stage, locus, allele, and factors is interchangeably used because the late 19th and 20s, we have the concept of the particles or factors, but we don't know exactly whether the locus is the physical position. So physical position is actually the concept brought by Thomas Morgan. We're going to learn in the probably the next session. Yeah, so that's the one. And the second one is the Pearson and Lee study, studied the parent and child height, and the correlation or the similarity is 0.5, which is a 50%. And then this is the representing the inheritance of the chromosomes, right? Because the segregation is the happen at the half at the each parent, right? Okay, then her question is the whether height is the traits affected by genetic factors. That was her question, yes. And how much affected by genetic factors? It's actually 20% according to these days heritability. So we're going to learn about the heritability in the later section, but if you use the heritability concept, then you can calculate how much the genetic factor is affecting on the trait, right? So that is the thing. And also some traits question asked that whether the traits are influenced by genetic factors or non-genetic factors, and can we formalized by some scores? Yes, it is, and that is heritability, right? We're going to learn in the later section. Okay, so and the third question is how much she should study about the mathematical concept. And then what one can, I mean, the one thing I can say that, I'm not put it into, the mathematical, whatever calculation in the exam. But I mean, if you try to understand what the formula means in this context, that would be great, right? So I mean, that has to be the things you should do. And regression is talking about the deviation, and the correlation is talking about the similarity, and that's the mathematical term. And the page 22, she asked about the genetic covariance, and the 13th page about the diagram. 13th page diagram is just mere, the correlation plot. So you can just, try to think about, Garten did some effort on the, looking at the correlation between parents and child, and that's all about. And other questions about the genetic covariance. This concept we're going to discuss more detail in heritability session. Okay, the last question is about the Galton study about the parent and the child height graph. The plot is the positive correlation, and Galton did the handwriting, and her question is, we should do these things in these days. Probably yes, in my laboratory. That's just kidding. No way. I mean, we can use the we use the computer, and we still represent the correlation using the line, right? But the things you have to remember is that the calculating the similarity is the heritability, and this heritability is studied using familiar relationship in 90s or 2000. So, I mean, 10 years ago, 10 years ago, we used the family to calculate the heritability. That time, people, they summarize the research using, like Galton's idea. But these days, we calculate the heritability using unrelated individual because we advanced over the time, right? So we don't rely on the familiar information anymore, right? But I'm teaching the heritability using the familiar relationship at the starting point. But these days we advanced our methodology, and this time people using unrelated individual for the heritability calculation. So that answer is yes or no. Okay, that's good. So if you have some questions, you can leave the public channel like Slack or Zoom. Don't send me the direct message, because I mean, what I want you do that is expose your question, discuss, and shape your understanding. Otherwise, you cannot learn anything from this course, right? This is the critical things in the university time, right? Because we have so many good lectures on the YouTube. So I mean, I'm sometimes curious about why we are going to university in these days, but I'm paid off by university. But the things we can do in these days is the discussion, right? So discussion and shaping your idea, and I'm mentoring your thought, and also give some guide on that. But if you only just take this course as, just get good grade, but I mean, I don't think it's not valid anymore. So who cares about grade in these days, right? So think about that situation and leave your question, and we're going to boost our idea in next session and next session. And if you don't follow this week class and no question, that's something bad, right? So please keep in mind that you have to come up with some questions, and otherwise you totally lost in some days. I mean, I would say so. Don't be shy. Yeah, open up your thought, and hopefully you enjoy the course, and we can discuss further next week. And some of our department students we're going to meet in Friday, and that time I will back to my own language, and this BS university rule forced me to do some English lecture, but anyway, I blame these things many times. Yeah, but it's recording, so I mean, I shouldn't whinge these things. Okay, so good, and no more questions. Okay, that's good, and see you next time.